What I'd like to do now is introduce Tim Wood, and wonder if somebody could have Tim enter the webinar. Dr. Tim Wood is an associate professor of pediatrics at the University of Colorado Anschutz Medical Campus and is director of the Biochemical Genetics and Laboratory at the Children's Hospital Colorado. He's involved in improving diagnosis for different inborn errors of metabolism, including MPS. And it's my pleasure to be able to have Tim give a lecture on the laboratory diagnosis of MPS. Tim, thank you very much for coming.
Well, thank you, Dr. Munzer, and thanks for allowing me to participate. Does my screen look okay? Well, I will get started. It's truly a privilege to be part of the seminar series. I've worked with Dr. Munzer for a number of years, and the information presented here, the group of people that are talking about these disorders, it's just it's an amazing group to be part of. Whoops. So I am a salaried employee of the University of Colorado Anschutz Medical Campus, and I do run a biochemical genetics lab. Other than that, I don't have many work conflicts. So what I'm going to talk about today is sort of the pieces of the laboratory side of making the diagnosis of an MPS disorder. We're going to start with some molecular testing, enzyme testing, and glycosaminoglycan analysis, and then I'm going to try to give you a summary. I'm going to tell you sort of how I understand this and what I know, and try to present on main concepts. But I hope you'll use the chat or the questions afterwards to either enhance what I talk about or to clarify anything that I say. So I do want to start with just sort of a broad concept, and that is that I'm going to say that your friend claims that their child is the fastest sprinter in the world, and they need your help in timing the child in a 100-meter dash to submit to the Guinness Book of World Records. And the question is, what timepiece are you going to use for this measurement? Are you going to go to London and use Big Ben? Are you going to use a digital stopwatch, a wall-mounted clock, or the old old-fashioned 1 Mississippi, 2 Mississippi, 3 Mississippi? Now, all of them will work. All of them will try to get you what the relevant amount of time it takes for this child to complete this task. But what the point I'm trying to make is that laboratory assays need to be calibrated to the clinical question that we're trying to ask for the patient in front of us. And a lot of the older methods that we've used were developed prior to next generation sequencing, prior to newborn screening. And so we may not have the data, or they may not have been calibrated to try to answer the intricate questions that we're trying to deal with in terms of, what do GAG levels look like in carriers? Can we tell a pseudodeficiency patient from a mildly affected patient, and things like that? So I'm going to try to weave that concept into the rest of my presentation. So the historical testing for MPS disorders really started with a clinical suspicion, or maybe a family history. They would do urinary GAG measurements. I'm going to tell you a little bit about those that had false positives and false negatives, depending on which MPS you were talking about. They also, in general, sort of lacked an analyte-specific quantitation component to them. If the urinary GAGs were positive, we often went to enzyme testing that, at least when I started, was done primarily on a fibroblast biopsy. So that's taking a little bit of skin, sending it to a lab. The lab grows it up in culture, and then we do the enzyme measurements on that. And then finally, and I know it's hard for people in the current times to believe this, but there was a time when we didn't even know the genes for all of the MPS, and we couldn't do sequencing. And sequencing was a research test that took months or sometimes years to get results back, if you got them back at all. So in terms of testing for an MPS disorder, it may take multiple visits, it may take multiple samples, and it may take weeks to months. Where we are now is when we think about making an MPS diagnosis from the laboratory side, or I guess the laboratory/clinical side, it's really a puzzle that we're trying to put together. And we want all of those pieces to come together to provide the information we need to make an accurate diagnosis. So you can start at any place. You can start with the GAGs, you can start with the enzyme. A lot of people now are starting with molecular, but we want all these pieces to point to the same diagnosis. And then we'll use the information that we gather to do treatment monitoring or other type things. So what I'm going to do in the rest of this talk is break down. I'm going to let Dr. Munzer and all the other really cool presenters talk about the clinical features of MPS. And I'm going to try to go through in a relatively brief time period to talk about the GAG analysis, the molecular analysis, and the enzyme analysis. So again, when I started, molecular analysis was the cool, neat thing, and it was often where we spent the most amount of our time. But now molecular analysis is really pretty straightforward, and I'm going to start here because it's really the I don't have a lot of information to give you in terms of unique aspects of MPS diagnosis. So most MPS disorders will, or the pathogenic or variants detected will be detected by standard molecular testing, such as next generation sequencing, or depending on where you are in the world, Sanger sequencing. Most of these conditions are related to missense mutations or missense variants that are easily detected by these technologies. I did want to point out that MPS 2, about 5 to 10% of the alleles, it should be percent of alleles, are due to a rearrangement with a processed pseudogene. So that's a piece of DNA that has sequence characteristics or sequence similarities to the functional gene, but is usually inactive. There's a specific rearrangement PCR that you can do, and there's a new paper out that talks a little bit about that. So if you are working on MPS 2 and your molecular analysis is normal, this may be something that you want to look at. The advances of long read sequencing for next generation sequencing will probably detect this, I believe. And keep in mind that for MPS 4A, about 10% of the alleles are outside of the coding region. So if you do an exome analysis, you might miss these, but certainly a genome analysis would detect them. Again, generally, large deletions, intronic changes, and rearrangements, while they've been described, are not super common in these conditions. When you do sequencing analysis, one of the things we're going to run into is what's called a variant of uncertain significance. This is a change in the DNA code that we're not exactly sure what it means. We're also understanding that some of these variants lead to a phenomenon called pseudodeficiency that I'll talk about. A molecular analysis is the best way to identify carriers if you're doing extended family studies. Molecular studies used to have a much higher cost than enzyme, but I believe that is coming down, and I know there's some targeted testing panels that exist that may even provide very low cost testing. And then I did want to add that as far as I understand, most prenatal testing that's done in the United States is going to be done using molecular techniques. So I think molecular testing for MPS is pretty straightforward. It's pretty widely available. And while the data, the variants of uncertain significance may wreak havoc, in terms of the laboratory side, it's pretty standard. So the second part of that diagnostic puzzle that I want to talk about is enzyme analysis. This is also called a functional analysis. And one of the things we need is we need the enzyme to not be degraded, and I'm going to go over that a little bit. So how does an enzyme analysis work? Let's see. Can I? Whoops. So a lot of these use what's called a fluorescent substrate. And what that means is you see I've put here white blood cells, fibroblasts, plasma, DBS. That's the sample type, and that adds the enzyme. The enzyme is going to act on the substrate, in this case, a fluorescent substrate called a 4-MU based substrate. And when that enzyme acts on the substrate, it's released, and that releases light or fluorescence that we can measure. In a patient who has the condition, they lack the enzyme, so the substrate is not cleaved, and so there's no fluorescence. The other thing that we use, the other technique that we use for enzyme analysis is mass spectrometry. And it's a very similar process. You have a sample type that adds an endogenous enzyme. The substrate is cleaved, but instead of looking for the release of light using fluorescence, we actually measure the specific change in the substrate, the molecular molecule that's been, or the molecular molecule that's been released by the enzyme. Okay, so how does enzyme testing work? Typically, you're going to collect blood or some sort of sample. You're going to collect blood that may be in terms of a dry blood spot. They may take the blood sample and spin it down in terms of to collect plasma. That's specific typically for MPS 2. And then finally, we may isolate the white blood cells from the blood sample, what we call a leukocyte test. So the enzymes are generally stable if you freeze them. And they're also generally very stable in dry blood spots. If we're trying to collect leukocytes and do a leukocyte assay, that's a little bit more difficult, and I'm going to describe that to you in a minute. And generally, if we're going to isolate leukocytes, we need to do that within 48 hours. You can think about it when you pull that blood sample, you're taking the leukocytes, you're taking the blood cells out of their nice, happy environment, you're putting them into an artificial environment, and over time, they'll start to lyse, they'll start to break down. Within about 48 hours of blood draw, you really start to see enough of that lysis, that breakdown of the white blood cells, that it really impacts the quality and the amount of the leukocytes that you get. So you really want to try to get those blood samples to the lab if you're doing a leukocyte assay within about 48 hours. And that's important, I think, to keep in mind that if you're drawing blood on a Friday and you are shipping it to the lab, you need to know whether that lab's available on Saturday to receive samples. Because if that sample is drawn on Friday and doesn't get to the lab till Monday, I'm sorry to say that labs may reject the sample based on the quality. And I know you've worked hard to get the patient or to get your child into the clinic. You fought with them while you had the blood draw, and then to hear from somebody like me, hey, sorry, we can't do it, is very frustrating. I get that. The flip side is I can't give you a quality result if I can't get good leukocytes. So once we get the leukocytes, we grind them up a little bit. It's not really a blender, but it's a good example of what happens. We release those enzymes into a test tube. You have a very competent technologist who performs the enzyme assay, trying to read the fluorescence using the slide I told you before. And then we have to do some calculations, and that's the last thing that I want to talk a little about, because we're expressing this enzyme activity as the amount of substrate that's released for the amount of blood, plasma, or lysate that we use. And so what can impact this calculation? And that's a little bit about what I want to tell you. So this enzyme activity, which is the thing we're trying to get to in the green in the center, can be impacted by several of the things that I just described. One of those is the isolation of the leukocytes. So if you're shipping this sample, either when it's really hot or when it's really cold, sometimes that sample will freeze or essentially get too hot during the transport to the lab. And so you can have quality problems either with how well the leukocytes work or how much hemolysis is there. If when they do the leukocyte, again, because of some hemolysis that occurs, you can get a little bit of redness or a little bit of hemoglobin that can artificially elevate the protein concentration. And because the denominator for leukocyte assays is the protein concentration, that a little bit of artificial extra protein in there can lower your enzyme activity. And then finally, a lot of what you're asking us to do in the lab is measure very low enzyme activities, and that can make it difficult. You're essentially asking us to measure very small amounts of something, and if we don't have the assay calibrated exactly right, or more often just due to small variations in some of the analytical parameters, that can cause variations in low enzyme activity. None of this is really going to change whether a person has activity in the normal range or very, very low. But when you have two children who have the same genotype, and we give you an activity of 0.7 in one and 0.3 in another, these are some of the small differences or some of the analytical parameters that can lead to those small differences, even though the patients have the exact same genotype. So these are just some of the things to keep in mind when you're trying to do comparisons of enzyme activities. So if we back up a little bit and we now have our enzyme activity, let's talk about the broader context of how this works on sort of a population scale. And the way we think about this is, let's say we have an enzyme that has an activity somewhere between 0 and around 80. And we have this normal range that if you were to pull a blood sample from anybody walking on the street, it would typically be between 6 and 76. And if we're looking at an enzyme that is associated with an MPS, we have a range that we note in patients with the condition. And I think it's real important to know that that's not always 0. There's a little bit of room there on the bottom in what we consider to be what's called the affected range. And then the way this works in the textbooks is anybody who's a carrier is here. They're clearly separated, and it's real simple. Well, as in most things in life, it's actually a little more difficult when you get in the real world. So here's a similar thing. Here's that activity range. Here's that normal range. Often the carriers are going to overlap with the low end of the normal range. Hopefully, when we've designed these assays, we've made sure that the range in patients is clearly separated from carriers and individuals who have no variants in the gene. And we use this hypothesis, we use this idea or this concept to do newborn screening for a lot of MPS. We're measuring enzyme activity in those dry blood spots, with this being the idea of how it's going to work. The problem that we ran into, and there were good publications about this, is something called pseudodeficiency. And I'm going to explain this to you in a minute, but the broad concept is, now we know that patients or individuals who carry these pseudodeficiency alleles, and I'll tell you more about those in a minute, can be from the low end of normal through the carriers and may actually go into what we consider to be the affected range. And that makes it difficult in a newborn screening context when you don't have the clinical information to determine whether a patient needs treatment or not. So when we get into newborn screening, there's two main methods that are used. One is MSMS, which is short for tandem mass spectrometry, and one is a fluorescent-based assay. And I do want to say that it's my opinion that both techniques can be used to diagnose patient with an MPS disorder. They have their pros and cons that I'll go through, but generally they both work very well. There are a couple of papers out there that suggest that the LCMS, I've sort of mixed my acronyms here, but tandem mass spectrometry, because it's a little bit better at getting at that lower end, it's a little more sensitive, maybe better than fluorescence to try to tease out some of the mild versus severe patients. And one of the really cool things about mass spectrometry is we can do a bunch of reactions in one single analytical reaction. We can measure a bunch of enzymes in one single reaction. And this is just a revolutionary concept emerging of a really cool technology with it's really changed how we think about doing diagnostic testing for MPS and all lysosomal diseases. We think about this as sort of a static, hey, these substrates, these analytical methods are out there, they're not really changing. But I do want to point out that Mike Gelb's group, and you're going to hear his name a lot, Mike has almost, I don't want to say single-handedly, but he has carried the flag for newborn screening of lysosomal diseases and this concept of using enzyme analysis and tandem mass spectrometry to do newborn screening for these conditions. And it's really powerful, amazing work. But Mike continues to advance and change, and he does have a new paper talking about a new substrate for MPS 2. So one of the key concepts, and maybe this may be the one thing that I hope that you really hear from this talk, is that remember when we're doing these measurements, we're almost looking, and we're doing enzyme analysis, we're looking for very low signal or almost no signal. And that's what we're using to make this designation that a patient has the condition. So what are some other things that could cause low enzyme results? And one of the first one is, and I've harped on this a couple of times, is poor sample quality and or some kind of laboratory error. If you have that poor sample quality, you're going to get a non-functional enzyme, not because of the genetics, but because of the sample quality. So one of the things you always want to ask for is whether a second enzyme was measured. And that second enzyme should be sort of, it should be unrelated to the enzyme that you're trying to test. And it's really just there. They may not give you a number, but they're going to tell you that that enzyme activity was within the normal range or within their QC parameters. And therefore the sample quality is okay. And we can use that. We can then use the results from the enzyme that you're interested in. I do want to point out, since we're talking about MPS, that there are a couple of non-MPS or MPS similar disorders that can give you low enzyme activity. One of those is multiple sulfatase. This is a condition where you cannot make a post-translational modification of most of or all your sulfatase enzymes. And so the sulfatase you're looking at is deficient, but also the sulfatase, there are a bunch of other sulfatases that are deficient. So when you measure a sulfatase, if it's good if that second enzyme is actually another sulfatase to rule this out. There's also another condition called mucolipidosis 2 and 3, and this is a process where you can't target the enzymes to the lysosome, and Dr. Munzer talked about where that what that organelle does. And so the enzyme is there and it's functional, it's just not getting to the right spot. And so you mainly see this in fibroblast assays. You don't really see this much in leukocytes, in leukocyte-based assays. But remember, if you're doing something in fibroblasts and you see a low enzyme activity, very rarely it could also be caused by mucolipidosis. And the way you test for that is you look for another MAN6P enzyme as your control, and you see that it's also low in the patient fibroblasts. So what I want to spend the rest of the time talking about is this idea of something called pseudodeficiency. Dr. Thomas is one of the first articles that I found that discusses this idea of pseudodeficiency. It's essentially where you get an enzyme activity level that's in that affected range that I described earlier, but it's in an individual who doesn't have the clinical phenotype. We came about this phenomenon when we would do carrier testing or we would do family testing in individuals with various lysosomal disorders, and we would find a parent, an unaffected parent or an unaffected sibling who had enzyme activities that matched or were within the same range as the patient. So because we're using enzyme activity in newborn screening, we're starting to find out how common pseudodeficiency is. I know I was one of the people that didn't think this would be as big an issue as it is. So I just want to talk a little bit about what pseudodeficiency is. It's not something that's just related to MPS. In fact, it was described first in a couple of other lysosomal conditions. These are a subset of disorders with known pseudodeficiency. And in fact, I think the list grows every year as we do more and more to find these alleles. I do want to talk a little bit about sort of the mechanism. So this is the ARSA pseudodeficiency. It's one of the ones that's been best described. And I want to introduce this concept of a complex allele. One of the reasons that, that this allele has such low activity is it's actually got two different changes. It's got a loss of a polyadenylation signal that decreases the amount of one of the main RNAs. Forms of the RNA, and it has a loss of one of 3 glycosylation sites that probably leads to some mistargeting. So it's this in-cis, it's two changes that are on the same allele. It's this haplotype that together leads to reduced enzyme activity. About 5 or 6 years ago, we started working on a document that talks about the measurement of lysosomal enzymes. Aaron Strovel put this together. And at the very back, almost as an afterthought, we did one, I think it was one of the first curations of pseudodeficiency alleles. And again, this is a table in the supplement. So if you want to learn more about or get a list, this is one of the first places to look. I also want to bring your attention to Dr. Goldstein, who's there at UNC with Dr. Munzer, and she's actually leading a ClinGen pseudodeficiency work group that's trying to get their head around this concept of what is a pseudodeficiency. What does this term mean? Are there other phrases that we can use that are more more patient friendly and maybe a little more accurate in terms of the phenomenon that we're describing? So look for that. Jenny's been given a couple of presentations at some of the national meetings. If you're interested, you can contact Jenny and become part of the pseudodeficiency work group. But hopefully in the next 3 to 5 years, this group and others will be trying to better define what pseudodeficiency is. So one of the little one of the other things I want to talk about again is this mechanism of pseudodeficiency. There's some early literature to suggest that this was sort of a lab problem and that it's not really a biologic phenomenon. And I'd like to put out there that I think we've got a whole bunch of these alleles, and that may be part of it. But as you start to read and study these alleles, I want you to understand that it's not just the fact. The early concept was that we're using an artificial substrate and not what goes on naturally in the cell. And it's the inability of these enzymes to bind and act on these artificial substrates that's the main problem. I think that could be a problem. It probably is a little bit of a problem. But as you read through these alleles, you find that there's reduced messenger RNA expression. I already talked about the change in glycosylation. There's reduced stability. There may be an overall reduction in enzyme activity, or like I used with the ARSA example, it may be an allele that's a combination of both. And so one of my friends and colleagues, Dr. Steet at the Greenwood Genetics Center, has several papers where he's describing what these alleles are. And he's putting them in a cell culture system so that he can study them more efficiently. This is hard work. It's expensive work. And it takes a little bit of time, but I really think it's something that as a community, we need to come together on and support Dr. Steet or others who are doing this kind of work, because it's really important to understand the biological consequences of these and develop a series of laboratory experiments that can help calibrate whether these are pathogenic or not. The other thing I want to say is that some of the new guidelines for variant interpretation are much stricter in the what papers can be used to define a functional to define whether a variant in these artificial constructs is functional or non-functional. And so we're really having to take not only this new look at it, but we're having to redo some older data because that older data doesn't meet the ACMG or other regulatory guidelines. So I do want to just show you this paper that's on the right. If you look, the wild type is on the left. And as you move across, what you see is decrease in what Rich calls the relative specific activity. So each change as you move along through here has, and this is a logarithmic scale, has decreases in activity due to various changes, and Rich and other papers talk about this. But I'd like to look toward the middle of the graph where you see you've got H82Q and L526P. And so if you look on the left-hand side, you can see where the variant L526P and H82Q, and those have about 10% activity. But when you put those two changes in cis or on the same haplotype, it further drives the enzyme activity where it's in the range or much closer to the range of what you would see in a mild patient. So I think this is really important when we start looking at our genotype data, not just to look at the variants that were identified, the VUS or the pathogenic changes, but also are there any pseudodeficiencies in there? And one of the things that Jenny's group is wrestling with is a guideline for what to report to make sure that the labs and the patient are communicating correctly about what genotype is actually there. Okay, so I will spend the last 15 or so minutes talking about GAG analysis, and then I'll be open to any questions that you guys have. So historically, urinary GAG measurements use the first two lines of this table. They either used a dye binding assay, what's called a total GAG assay, or you might see it in the patient's chart as a DMB assay because that's the dye that was used. It was a quantitative assay. It was standardized across labs. It was widely available. It was low cost, all things that we like. The limitation was that quantitatively, it didn't tell you which GAG species was elevated, and that the dye did bind some, did have some non-specific binding, so that false positives, I don't know if I should say they are common, but they did occur, and that false negatives, unfortunately, were pretty well documented. So what you would then move is to what's called a qualitative GAG analysis. This was something that was done. Typically on either a thin layer chromatography or a gel electrophoresis. It typically wasn't quantitative, although you could work to make it that way. It did identify which GAG species were elevated, but some of the GAG species were more difficult, like keratin sulfate. Was more difficult to detect using, at least for a lot of labs, it was difficult to do. And it did require a larger sample than the next technique I'm going to talk about. So when I started, this is what we were doing. We were doing a dye binding assay and we were doing a quantitative GAG assay. And we were trying to put these two tests together, typically in a urine sample from a patient, to try to identify what GAGs were there. And Dr. Munzer already talked about whether when you see the GAG species that was elevated would help point you in the direction as to what condition the patient has. So dermatin and heparin got you a subset of conditions. Just dermatin would get you another set, just keratin and heparin in another. So what happened is about 15, maybe 20 years ago, we started to again use this technology that I mentioned earlier called mass spectrometry, or in this case, tandem mass spectrometry, to basically combine what the dye binding assay and the qualitative assay could do. What we could do here is we could actually identify and quantitate, put a number to each GAG species. It was highly sensitive. It was able to work on multiple sample types, which is going to be hugely important. It was a little more useful in treatment monitoring, although somebody like Dr. Munzer may argue with me about that. And the amount of sample that we needed was an order of magnitude less. So from here on out, we're going to talk about mass spectrometry. If you're going to do these measurements, I really encourage any physicians, families, to use a technique that uses mass spectrometry. You can use dye binding, you can use the qualitative assays, but mass spectrometry is much more widely available now, and it's generally just a better test. So from here on out, everything I'm going to talk about are mass spectrometry-based methods. The good news is that they all do the things that I say here in the benefit. The bad news is that they're all different analytical methods, and we're using typically, hey, the gags were elevated. And it's important to know which gags we're talking about and how they were done so that you can sort of calibrate or so that you can compare between patients and between studies. So I'm going to talk to you about something called an intact gag analysis, or Mike Gelb's group coined the term internal disaccharides. And so I'm going to present to you the three main glycosaminoglycans that we're talking about: dermatan sulfate, heparin sulfate, and keratin sulfate. And I've put here some other names, this D0, little a4, D0, A0. These are some of the names that you're going to see as you read the literature on MPS. And so I want you at this point to start associating those molecules with the parent molecule that we're used to talking about. So I've tried to represent the disaccharide as a repeating unit of a square and a circle. And so when you look at these molecules, they're long repeating units. They may have modifications to them, such as being sulfated, and I've used that as a little designation of an S here. I want to come back to that. Keep in mind what those S's mean. So typically what we do is we take a urine sample, we'll treat it with a chemical, or we'll treat it with a series of enzymes to digest these long polymers into those circle and square disaccharides, or 2 units. We then separate and quantitate using this super cool method called an LCMS. And what we do is we try to measure the amount of these disaccharides using this instrument. And so in normal, if abundance is higher up on the scale, a normal will have a certain amount of each of these disaccharides. Somebody with an MPS disorder who has a block in the ability to degrade these and therefore has elevations in certain GAG species will have higher amounts, higher numbers of these disaccharides. And we then use that to go back and say which MPS condition they might have. So this is the middle example is a patient who has elevations in dermatan and heparin sulfate. And then for a patient with MPS 3, typically the dermatan and the keratin will be normal or near normal, but you have a significant elevation of heparin sulfate. So this is one method called the internal disaccharide method. One of the other things that was described is something called a non-reducing end. And the idea, or at least the way I understand this concept, is that the digestion of these long GAG polymers is unidirectional. It occurs from one end called the non-reducing end. And it's a series of enzyme steps, which is shown here, and Dr. Munzer showed you a similar diagram. And what you do is when you look on the left-hand side, the structure that's sitting there at the end is unique to typically one MPS. And so what you'll have in a patient with MPS is GAG fragments that are enriched on one end with one of these unique structures. And what very smart people came up with is that, hey, if we could look at the non-reducing end of the GAG molecules, we could relate that back to which MPS condition. There should be a specific structure that's specific for each MPS. And so that's a test. I believe the diagnostic name is called the SensaPro assay. But essentially, what they did is they came up with a whole series of enzymes and steps that not only releases the non-reducing end, but also looks at those internal disaccharides. And so this is a really cool test because it looks at not only what the non-reducing end is, which is disease specific, but it also allows you to look at what those internal disaccharides are. So it's a test that gives you both. It's a complex test, and it's not as fast as the next one I'm going to show you. But I want you to get the idea that a non-reducing end is a molecule that is specific for an individual MPS condition. So during the time that they were working on that non-reduced, the SensaPro assay, Maria Fuller from Australia had identified that there are these what's called endogenous non-reducing end. There are these free molecules floating around that are similar to what they described in SensaPro, but you don't have to do all of the digestions. That you can just take the sample, look for these molecules, and if they're there, they have an MPS. And so again, Dr. Gelb calls these endogenous NREs, and this is becoming what's the dominant method for GAG analysis. So here are some of the structures that are on the right. This is from a paper in 2019. It shows you which is the structure and which MPS they're specific for. Here is a table from that paper talking about, again, the molecules and the relative amounts. And then here's an example showing that they are receptive to enzyme treatment. So this is just a table that if you're able to go back and look at this, what are the different GAG analyses via LCMS or tandem mass spectrometry? Chemical digestion and enzyme digestion are fairly similar. One of the nice things about enzyme digestion is that you keep those modifications that I talked about. And so you'll probably hear a molecule called, I think it's D2S2, as a very unique marker for pathogenicity in some MPS. I believe you have to have an enzyme digestion to pick that up using the first two methods. I talked to you about the SensaPro, and then there are a number of labs, and where everything is going is to that endogenous method, what I call it here GAG biomarkers, and there are a number of different techniques that you can use. So everything I've talked about at this point is dealing with doing GAG analysis in urine. But once we, there's two papers I'm going to talk about that came out of Dr. Gelb's group. Both of them do really nice at explaining these molecules. And this is a really nice table that puts together everything that I just talked about, the accumulating parent GAG, the internal disaccharide method, and then finally the NRE biomarker. So once we started doing newborn screening for MPS, and we started realizing there were going to be these pseudodeficiency alleles that drove enzyme activity that gave us false positives, we needed to come up with some way to identify whether the patient was going to be, or try to identify whether the patient would have the condition or not. And so at the time, what we were doing, if you think about your diagnostic algorithm, is we were trying to do, we were bringing the patient in, we were drawing a blood sample, we were collecting a urine sample, which is super hard to do on a little baby. And then we were sending that off to a clinical lab, and it took at least a month or several weeks to do. And then some very bright people in several different labs, Dr. Tomatsu's lab, Mayo, a couple of other places, came up with this idea that we could actually measure the GAGs in dry blood spot, and we could do that from the dry blood spot that was used to do the initial newborn screen. And so these are some internal disaccharide measurements that I believe, I know the one on the right came out of Mayo, showing that actually GAGs were elevated in dry blood spot samples from patients with MPS. And so these are two papers from Dr. Gelb's group. I think they do an amazing job just talking about the different methods. And I really think it's kudos to Dr. Gelb's group, because what they did in working with the MPS Society is they collected a whole series of dry blood spots from MPS patients as well as normal unaffected individuals. And they took the same spots and they compared them across several methods. And what came out of this work is that while internal disaccharide methods are good at identifying patients with MPS, these endogenous NREs, the Maria Fuller method, is really better at it. And it's better because there are smaller false positives. So they did an initial study with MPS 1, they did another study with MPS 2, and then finally at the bottom, they looked at a series of patients with a variety of MPS, and all of them showed the same thing, that these endogenous NRE markers have the lowest number of false positives. And so what you're seeing.
So Tim, let you know you have a few minutes left, so just be aware of time.
Yep, thank you. So what you're seeing now is a whole series of papers that bear this out. This is a nice one again from Dr. Gelb's group. And I think really where the field is moving is that anytime you get a positive on a dry blood spot, we're then taking as a second tier test to measure endogenous NREs. And hopefully that's going to significantly reduce any false positives that we have, because patients with an MPS disorder will have a significant elevation. So in terms of second tier follow-up for MPS disorders, in terms of newborn screening, when we started this, we could do molecular testing, a blood-based enzyme testing, a urinary GAG analysis. Again, the last three the last two required a second blood sample or a urine sample on a baby. But I think where we're coming to now is that a dry blood spot GAG analysis is the best follow-up. You can use one of two GAG methods, but generally most places are moving to the endogenous NRE. So let's talk about a couple of summaries. GAG testing methods, molecular testing, it's pretty straightforward, but you're going to identify some VUS. You're also going to identify those pseudo deficiencies. An enzyme analysis is a really good functional test, may require a different sample type. And we've realized that we have these pseudo deficiency alleles that drive the activity down. And so we need a way to deal with those. That's either the expression work that Dr. Steet does and others do, or it's a corresponding GAG analysis. And it's the mixing of all of these that we're hoping to put back together to make the diagnosis of MPS in a patient. These are just a couple of new articles that I came upon. The one on the left is using EHR, a medical record, to identify patients who are homozygous for pseudodeficiency. And I just pulled one of the relevant lines, is that we found no clinically relevant differences between cases and controls in patients that were homozygous. So I do think we've got some good data here to suggest that certain genotypes are not going to present. So a really nice paper on MPS 2 about novel insights into this one variant. I bring up this article because these individuals who don't have symptoms of MPS did have mildly elevated GAGs through a couple of different methods. And then finally, I think it's super cool. Taiwan has done so much with MPS, and now they're starting to implement newborn screening for MPS 4. Again, the overall concept, try to put the different pieces of the puzzle together and know that the assays that we're using may not have been calibrated to answer your specific questions, but I hope you'll contact myself, Rich Steet, various lab directors, so we can discuss patients and all work together to make a diagnosis. I will stop there, and thanks for your time.
Thank you very much, Tim, for that really nice overview in terms of the challenges. Just to while they wait for some questions, what's the preferred sample for a child with MPS? What would you prefer? Urine?
Yeah, that's
dried blood spot. And can you do everything off a single dried blood spot in terms of diagnosis?
so Yeah, we're to the point now where definitely we can do the molecular and the enzyme off of a dry blood spot. I think more and more labs are going to start measuring the GAGs in a blood spot. I'm a little more conservative. I think there's a lot more data out there about the urine GAGs. And so I would love to see doing blood-based enzyme and molecular, and then do a urinary GAG test. But there's a lot of good supporting data out there to say that you can do it all out of blood.
And as you appreciate, it's much easier to ship a dried blood spot sample than to ship a frozen urine,
Yep.
especially maybe not in the US so much, but other parts of the world, that's a significant issue.
Yeah, and so I think that's the really, I'm so glad you bring that up, Dr. Munzer, because I do think you have to think about your testing in the logistics and the patients that you're seeing. And it's nice that we have all these options. So if you're seeing that patient on Friday, collect a dry blood spot, send it to the lab. You're exactly right. You can just put that sample in the mail. It gets there Tuesday and Wednesday, and you remove all the problems with the leukocyte isolation. Yeah.
So a question in the chat, do all states use MSMS in their state labs when testing for MPS 1 and 2? Also, do states do secondary testing? They say enzyme level to call out false positive before patients are notified.
I won't use the word all, but there are these two methods. There's a fluorometric method, and then there is MSMS. And so for determining low enzyme activity, I think those two techniques do a pretty good job. It's some of the nuances about low enzyme activity that that may see a difference between the two. Do all states do secondary testing? So the secondary testing or the follow-up testing that I talk about would be those dry blood spot GAG levels. And I think most states are starting to send that to labs like Revity or Mayo. But I think whatever state you're in, you would need to ask how they do that. But I think the goal is, and I look to Dr. Munzer or somebody to agree with this, is that anytime there's a low enzyme activity, we're reflexing on the original spot to a GAG analysis so that we minimize the amount of patients that we're calling back. Would you agree with that, Dr. Munzer?
Absolutely, Tim. That's certainly the way to go. And I think there's studies supporting that. So the last question I'll let you take is PerkinElmer the only clinical lab available for the Fuller method? You alluded to Mayo doing it too, and not endogenous. Are there other labs doing that besides Revity and Mayo?
I believe I believe that's a little bit of a nuanced question because it depends on what sample type you're doing. I believe CHOP does the endogenous NRE method. I believe Greenwood's close to doing it if they don't. I don't know whether they do it in blood or whether they do it in urine. And I think it's Mayo and Revity, or may just be Revity, that does the endogenous NRE as a main lab or a big clinical lab in the US. But hopefully somebody can talk about that.
It's a comment from somebody. Mayo now has the non-reducing ends. We call them endogenous biomarkers for MPS 1 and MPS 2, focus more on newborn screening versus clinical testing. So thank you, Tim, for
Great.
that. We're going to take a break for a few minutes to come back at 10 after. Thank you, everybody. See you soon.